It's live.
Well, there we go. Good morning, everybody. Welcome to the Faika broadcast. I'm your host, Carl, and your lovely co-host, Naki, is not with us this morning, and we're not broadcasting from the Molly living room. Naki is, first she kicks cancer's ass, and now she's out there kicking COVID-19's ass too. So thoughts and prayers out to the Molly family. If it were a football game, they're pretty much taking a knee, we believe, and running the clock out. They're doing just fine. So I don't know if our guest today is a 49er fan, but I usually wear these obnoxious Steeler outfits just for Naki, because I know that she loves them so much.
Not too bad. I don't really follow football that much, but any team that has Samoans on them, I am there.
Well, I think I'll just have to wear it again next week, because if I'm not irritating you enough with this obnoxious outfit, then I have to get the effect out of it. So I'll wear it again next week for Naki. Our guest today. So normally we don't have guests on Sunday because of COVID-19. We are out of our physical studio, and we've been in Naki's, we've been in her dining room, we've been in her kitchen nook. But we do actually have a guest today, and he doesn't know this yet, but he's actually our co-host too. So he's going to answer some questions, and he's also going to help us get through the show. Our guest today is Dr. Andrew Patiai Young. He's a Samoan investigator at the biotechnology company Dren Bio. Dr. A Young was born and raised in the village of Faleniu in American Samoa. He received his Bachelor of Science degree in molecular biology at the University of New Mexico. His discovery of the function of a novel protein complex involved in intracellular communication led to several peer-reviewed publications and earned him a PhD in biological chemistry from UCLA. He then pursued an additional 4 years of research as a postdoc fellow at the company Genentech, where he identified a novel gene that regulates inflammation in disease models of rheumatoid arthritis and skin allergy. And I think that's the big thing that we want to talk about today is inflammation. I mean, I don't think people understand how important the control of inflammation is with all diseases or most.
Yeah.
So talking about that, we're gonna get into that, and there's no better time to have a Pacific Islander scientist, doctor that has worked on vaccines than right now, because obviously, everybody's got questions. Everybody's, it seems like it's a political thing. It's not even a medical thing these days, that everybody is a vaccine expert, I see on Facebook now. So it's good to have a true expert. How's your week? How have you been doing with the vaccine news? Is that big news in your world, or is it just us on Facebook?
No, it's exciting news, and I think we should all celebrate this milestone. I mean, this is I feel like the fruit of Operation Warp Speed, right? And I think we've heard from Pfizer that apparently they didn't take any money from any support from Operation Warp Speed, that they did this independently. But Moderna was part of this collaboration with the government, and the results look exciting. I actually don't know. I mean, there's a lot that has been going on in the past couple of weeks. So I'm not familiar with whether they've defined sort of a distribution strategy or when that's going to happen. But personally, from sort of a scientific, from sort of research perspective, this is great news. And just to clarify, the company that I work in, we're a very small company. We're 10 people, and when this happened, when COVID came into our lives, all companies, big and small, wanted to contribute. They wanted to be a part of the discovery process, but also to eventually get a drug. So we were also interested in getting into that. So I did some of the early experiments in the lab to see if we're even we even have the resources or the capacity or the right assays in place to get into that space. As I mentioned, we're a startup company, so unfortunately we just did not have that capacity. So we stopped working on that, but thankfully so many other companies are, and we have some success now. So
we spent a lot of time with you and Dr. Samoa on the Koviki Talk series, talking about clinical trials and really everything from a scientific standpoint. And it seems like, and this is from somebody who's obviously not well educated in the medical field, but it seems like the more information gets out there, the more that social media just loses it and goes off on these tangents. And I just got to think that the scientific field just shakes their head. Do you even do things like on social media, even, I don't know, CNN and Fox, are those things that the scientific field kind of looks at and shakes their head, or it seems like it is to me, and I'm not of that knowledge.
No, I completely agree. And I don't know if we're Facebook friends, but I actually haven't even posted anything about the vaccines. I mean, I'm read, press releases and all these, so many articles, and even in the news it's reported, which is great. I think it's been a dark cloud for the past almost a year now, right?
Mm-hmm.
We're heading towards the end of the year, and also we're heading towards, we've just gone through Thanksgiving, so I think it was some good positive news for the world. But all these press releases claim that there's, it's 95% efficacy and protection. But I'm waiting for the papers to, I'm waiting for the articles to be published, and there's always that delay, right? So normally, when you have data, they go through presentations, they're presented at conferences. So in fact, I'm actually attending a conference right now. So it's going through this weekend and next week, and this is the hematological conference. It's a very big conference looking at leukemia, hematological diseases. And I noticed that there's some of the COVID abstracts are in the conference, and I don't know if there's going to be any presentation on any interim preliminary clinical data yet. But I'll take a look and I can definitely let you know. But we're, most scientists, and I'm sure Dr. Samoa as well, we're waiting to see the actual, the actual data, the figures. But as a, just to point out that I think this is preliminary data after the first injection. I think this is collected 1 month after the first injection, I believe. So once all that data comes out, it'll be, we'll have more confidence in the, but I also don't want to disregard the work of scientists in those companies, and also the Data Administration Board, the NIH, and the FDA, that sort of looks at this data and determine whether it's worth getting out.
Do you think there's anything to be learned from if other countries move forward first with any of the vaccines that'll become available for us? Do you think is there anything to be learned from other countries doing it, or do you think it's so close in rollout that it really isn't going to give us any better idea of the efficacy?
So, it sounds like, and I apologize, I haven't looked carefully in what's going on in the news, but it sounds like Europe, or what's it, the UK, that is actually planning to
I think so.
or have approved for emergency use and distribution. So again, I'm not super familiar with sort of the research governance in the UK, and I mean, I'm sure there are excellent scientists and clinicians there that would most likely have reviewed the data. So yeah, it's hard for me to comment on at this point.
That makes sense.
But if it does, if it does get approved, and it gets, it's distributed to citizens, then that also itself becomes becomes a trial on its own, if you know what I mean, right? So we have to take a close look at how the general population responds to that. You know, eventually there's going to be a vaccine, and there are going to be people who get it first, right? So, and that data is essential to see, do we continue the process of vaccination or not? Or do we take a little break and sort of review how are people responding to it? So, yeah.
I saw a UN press release that was basically kind of, they were saying that as the vaccines roll out, that the powerful countries can't trample on the impoverished nations on a race to use the vaccine. So basically saying to make sure to look out for, poor countries and make sure they receive it too. Is that something as a scientist when you work on, like you're really a step of creating the product, but is that something that as a doctor and as a scientist that you think about in, like when we'll get into the novel protein that you discovered and worked on, is that thought down the line of how does it get dispersed, or are you really looking at like, I need to do my job to get the product, and it's down the line, it's their job to make sure it's distributed?
That's a fantastic question. That is something that's in my mind. You know, as a Pacific Islander, as a person of color, and just knowing, just being in these meetings and being the only person of Pacific Islander descent. And I know this from Genentech. I've been there for 4.5 years, and there's not one Pacific Islander in research. And I met a couple in sort of other aspects of the company, but not in research. And, Genentech covers so many different diseases, right? So, and I think, and a lot of the indications are determined based on what is it in for the company, right? But also from sort of the scientific perspective, the scientist who leads these projects, right? Like there's also bias in that, right? They look at where they get the most data. Especially when looking at diseases, we always look at what are the genetic variables, what are the genetic underlying factors, so what mutations are prevalent, that kind of stuff. And so that depends on data that's available, right? So a lot of this data is coming from, European, of European descent or Asian descent, but not aggregated down to South Asian or South Pacific, right? And that's because there's just no such data available. So for me, I try to look at those data. I try to find those data. But if we cannot find that data, then we work with what we have. But we try to translate it to, whoever has a disease can be considered for these trials, for eventually for the medicine. So, to answer your question, that is something that I try to bring to the table. But it helps when we have that type of data available, right? And so the question is, how can we get that data available? So we have to work with local governments, local hospitals, establish research infrastructure in local governments to help not just educate, but to get our people involved in sort of getting those valuable samples published, but then eventually have those be a part of the pipeline where we think about drug discovery and develop drug development.
I think that's you bring up a really good point, and you and Dr. Samoa have both said that we need our people at the table that make decisions. And the further up, so to speak, that we get, it's almost like overrepresentation, right? We make up such a small percentage of the population, but if there's 9 decision makers and where there's one of us there, that's a huge representation for a small population. So to the kids out there going through the education, looking at Dr. Young and Dr. Samoa and all the like the clinical psychiatrists and the Pacific Islanders out there, those are people at the table where they're making the decisions. So hopefully there are people out there like you that are looking at that aren't just segmented or kind of in a silo with their piece on the conveyor belt. They're looking down the line and hopefully trying to affect change down the line.
Yeah, and sorry, just
No, you're
to
good.
touch on one point. My start in science started, this began in Samoa. And, many people who I meet, who, I come across, they didn't think that I was born or educated in Samoa. They thought this was something that developed, from being educated here. But no, this started back in Samoa. It was in 2002, I believe, or 2003, that my high school principal, Ruth Tofinga, she's currently the Department of Education Director in Samoa. And she started at Tafuna High School. She started a class called the Specialized Science class that really focused on training students how to do research. And there was a talented and my great friend and mentor, high school teacher from India, Dr. Rose, and he was the one who taught the class, and we focused on how do you develop a research question? How do you do experiments, right? And that opened up, my mind about, oh wow, science is more than calculus or physics. It's more than the dry stuff that you have to memorize, and no one has fun. I mean, I hate memorization. I never liked those classes. I mean, I took it because I had to take it and I had to do well in those classes, right? But actual science, actual research is actually quite fun. And we had the Kobiki talk, and you guys had, I was explaining sort of like how we think about vaccine, how it's like a game to me, right? It's like a puzzle. And I think the more students realize the true nature of basic science, I think we could get a lot more students involved in science. And we recently, last month, so at the end of November, American Samoa had his annual science symposium competition. And this is where all the students who have done research and they've written research papers, kind of like the one that I published, they're doing it. Okay, so they write a whole research paper and they submit it. And for the past 5 years, I've been a judge on those papers. So they send out the papers to scientists in the U.S. And I think there's one guy at the NIH who's also fortunately helping out and reviewing the papers. And Dr. Samoa at one point helped out review. So I had recruited him to help out, I think, a couple of years ago. So this year we got 18 papers, 18 excellent papers from students, and they're doing this stuff, and so. And they're having fun doing it, and I hope that more and more students get to do this. And you can tell from the quality of the paper how much progression. There's usually students start early in their high school career, and they continue on the same project throughout high school. And that's how research happens, right? You start with something and you do this, you progress. And you can see the progression. By year 4, you see the same student again with the paper that I would say we can publish this now, So that's and it's education. It's just about giving them the opportunity, and they're getting this in Samoa. I just hope that, more teachers get involved, more students get involved. So that was the start for me. And it's great to hear that students are getting in on this as well.
I think it's a great point that where you start, there can be a misperception of, where you were educated or where you got your start. I remember Naki and I had, we were at Tangato Bakery in Salt Lake City, and we ended up talking to a Tongan programmer, and he felt like there weren't enough outlets for Pacific Islander artists, so he created an app on his spare time, and he's a programmer, and so it was pretty easy for him to do. But he was, after the interview, he was talking about he really was frustrated with the lack of Pacific Islanders that are in math and science. And the way he put it, it was really interesting to me that he said, we're so good at that. You would think it would come natural, but it just isn't a major focus, like other education paths in schools for Pacific Islanders. But the way he said it, it was just with the way he felt like it's just so natural for us. We're good at math and science, but we don't end up in a lot of fields that we could, through distractions or other things. But I took away from that I'm Pacific Islander, and I wouldn't have said in that way of like we're really good at math and science, but we just don't. It's not something we focus on. So hopefully that what you're looking at, what you and Dr. Samoa are looking at, are the up and coming next generation of students in that field. So is that were you in high school? At that point, did you know kind of, you had talked about that's the path that they kind of stay on.
Yeah.
Did you know in high school where you wanted to be?
So no, I didn't know. So I wanted to be a medical doctor. But as you know, we grow up being fed that, right? Like, the only careers out there are a lawyer, medical doctor, rocket scientist, whatever. But that was, let me take a step back. One of the reasons I joined the class was because more, selfish personal reason. I knew that they had these like, great prizes. If you win, you get to go on a trip to Hawaii, right? And I had never been on a plane before. And I always, I always wanted to be on a plane. I always wanted to leave Samoa. And my parents, who played, such an instrumental role in our lives and education, they told us that if you want to get on the plane, you pay your own ticket, right? So, and that was, that was like, okay, that's an opportunity. I could do it. But also, I was lucky enough there was a program right after ninth grade, and I had an English teacher. Her name is Veronica, Mrs. Am, we call her. And she taught my English class. It was a great class. I felt my English improved so much after taking her class. She left in the middle of the school year to get a job at the college. And this is important information, because at the college, she led the Upward Bound Science and Math program. I don't know if you're familiar about this program. It was a program, I think it still exists today, but it was developed many years ago as an effort to push more minorities into STEM. Right, we were always talking about getting more minorities into STEM, and so this was a particular program by the NIH to get people, minorities, into underrepresented individuals into math and science. And so when she started that job, she found that there's a program in the US where anyone from Samoa can go. It was just not, for some reason, the application was not distributed to students. So she called me and said, oh, there's this, it's due tomorrow. Are you willing do you want to just fill this out and give it a try? So I got into the program, and that was my first time coming to the US, and I came alone. I was just 13 years old. I came alone to San Diego, and I attended the program, and it gave me that first perspective of, I learned biology at the college level. I learned, calculus at the college level. That already provided an edge, right? So now I'm going back to Samoa, to high school, to take a biology course that in a way I've sort of like pre-studied or prepared myself for that course. It was such a breeze to go through that course. So instead of taking that course, they put me in this specialized science class. Now I can just focus on doing research. So I've had many different paths I mean, science was always pulling me towards, towards itself. But when I got to college, I was just like, I didn't have a sense of like what a scientist is. I was doing science work, but I didn't really meet a scientist or what their career is like, right? So I was just focused on being a medical doctor for some reason. But when I got to college, I decided to join, to earn some money. There are these programs in college that like, oh, you could do research to get a stipend. So then I interviewed for one of these programs, and the first question is, what do you want to be after college? And I said, oh, I want to go to medical school. I want to be a physician. I want to be like Dr. Samoa. And they said, oh, okay. Yeah, no, we can't take you. So just like that, and they said, yeah, no, this program is only for targeting students who want to do PhD. And they have a quota to meet, right? So they have to demonstrate to NIH, to federal funding, that they've gotten students into PhD programs. So every time someone joins and goes to another program, it takes away from the point of the program, and so they lose funding. So then I went to the second program, and of course, guess what I did? I said, oh, I want to get a PhD. So then they took me in right away, and I started doing PhD research. That changed it. That was it for me. I knew this is what I wanted to do. And since then, I was just on the track to doing basic research, getting a PhD, the rest.
That's amazing. That's amazing that you are where you are, you love where you're at, and it wasn't the original path that you had drawn out for yourself. You're listening to the Faika podcast, and please share out with everybody. We're listening to Dr. Patiai Young talk about his journey to his PhD. So quick question before we jump on the research that you've broken. New Mexico State, was it University of New Mexico? How did you end up out in the desert?
University of New Mexico. And, it's a funny story because, when you take an SAT, right, you start getting all of these in the mail, right? All of these like advertisements from many different schools. And, my parents didn't make a lot of money, like most parents, like most families in Samoa. And I just applied. I just applied to as many schools, and I didn't even, how in the US, students get to go visit, right? They go on these trips, they go visit colleges, but also they do research on like what exactly kind of college you want to go to. For me, my experience, my knowledge of colleges and universities was limited to what I was getting in the mail. So all of those universities, that was my, and the internet was slow in Samoa, so I didn't get to like Google and like find out all the different colleges or what the reputation, what Ivy League schools are and whatnot. So I was applying to a number of colleges, and New Mexico, University of New Mexico in Albuquerque, offered me the best scholarship of the ones that I got accepted in. Because I was not going to let my parents pay for, and I knew college was expensive. So they offered me an academics, it was a partial scholarship, academic scholarship, and I accepted the offer. But during the same time, I don't know if you're familiar with the Gates Millennium Scholars Program. So the Gates Millennium Scholarship, it doesn't exist anymore, unfortunately. I think they stopped like 2 years ago. But this was a program by the Gates Foundation. And their goal was to, in a period of 20 years, every year, they offered a scholarship to 1,000 students in the US, the territories, everywhere. And the goal is to get, and it's only for disadvantaged students and minorities. And it pays for everything. Okay, it paid for, you know, your room and board. Basically, what they do is whatever the financial aid doesn't cover, so you get your financial aid, and then you have the remaining debt. And then the remaining debt is what they take care of. And they do that for 10 years. So what I mean is they pay it for 10 years of my education, right? So it went beyond college, it went to my grad school as well. So not only did I get this partial scholarship to go to New Mexico, but later on, I got the news that I got the Millennium Scholarship. And with the Millennium Scholarship, I could have gone to any school, right? Because they'll pay for it. At the time, there was no cap. I think later on in the year, they capped it to like maybe 50 grand. So I was basically covered from college and then throughout my grad school career. So I totally lucked out, and I'm glad I also went to University of New Mexico, because there's some amazing people there, amazing programs led by Margaret Warner Washburn, Dr. Maggie. And she loved Samoa. You know, she did a sabbatical back in the days in Samoa. So she in Western Samoa. So she has a special place in her heart for Samoa. And that's how I ended up in New Mexico. I'd never been there before.
I was going to say, I used to have New Mexico in my region in my 9 to 5. It gets cold in New Mexico. Did you even have a coat ready that was built for that kind of cold?
Nothing ready. And, you know, I started in the fall, right? And I remember the plane, you know, descending, and it's just desert everywhere, at least where the airport was. It's just like miles and miles. And I was like, God, what did I get myself into? But then what people don't know about New Mexico, at least for Albuquerque, is that it's high elevation, right? And so they have all 4 seasons. They have blizzards, and I remember my 4 years there. 2 of the years they canceled all the fall exams, final exams, because of the snowstorms. So whatever grade you got going into the you know, there was no virtual, none of that stuff back then. So whatever grade you got getting into the finals, that's your grade. So it's all good. But yeah, it was my first snow, you know, white Christmas there as well. So it was great.
I bet it was fun.
Yeah.
All right. So again, if you're on Facebook, share this out, because we're going to go over some really cool information with Dr. Young about inflammation. And I think that maybe you can give us a quick run through of, because I don't think I didn't until a few years ago. I didn't understand how damaging inflammation can be and how it plays into a whole bunch of different diseases or illnesses. So maybe give us a rundown of why controlling inflammation is important.
Yeah, definitely. So inflammation is typically the start of like a start of a disease, right? Okay, for example, in my case, we were looking at skin allergy reactions as well as rheumatoid arthritis. So inflammation is sort of like the initial insult. You have let's say you have a cut, and then you start getting all the swelling on sort of the side of the cut. So that's the start of inflammation, or, you know, another term for it is vascular leakage. Basically, your vasculature or your blood vessels, they start to leak out, right? So they start to leak out blood because the vessels start to swell, and the vessels are sort of, you know, you have cells that are connected to each other through junctions that keep the sort of the integrity of the blood vessels. So that starts to swell, they start to break up, and you have, you know, immune cells, they start to leak in. So when they enter into sort of the beyond the blood vessels, they secrete, you know, what we were talking about in Koviki talk, they secrete, you know, cytokines. You hear about this like cytokine storm, so that's massive inflammation, right? So if you don't control that, that becomes chronic, and it deteriorates the tissue, because that needs to be, there's always a balance, right? So you want to induce inflammation so that you recruit immune cells, to help with an infection. At the same time, you don't want them to be there the whole time because they keep repeating the process, and that eventually damages the tissue. So that's why it's important to control inflammation. That's sort of an example, a single example. And then there are like, yeah.
One of the and hopefully you give the right answer, and my kids are going to kill me. When they would sprain their ankle, I would get a big bucket of ice water, and as quickly as possible, I would have them put their foot in the ice water, not use the ice pack because you might miss, the ice slips. It doesn't, you don't get the area. So they would put their feet in these buckets of ice water to keep the inflammation down. Was that the right thing to do? Because they hated it.
Yeah, so that's how, so usually what ice does, it acts as like a constricting agent, right? So it just narrows the blood vessels. So it narrows, it reduces the blood flow to sort of that region. And so that slows down sort of, the rush of like, blood and immune cells to those regions, and it slows down inflammation that way. So it just constricts the blood vessels.
There was a lot of tears shed. They wouldn't want to tell me that I sprained something because I was like big on the ice bucket. So I'm glad that I didn't put them through that.
Ice is good.
For nothing.
It's funny. My mom also just recently had a knee replacement surgery. And there was a lot of swelling, and that was, she was on pain medication, but the doctors also said, just like wear those compression socks, right? Again, it's all about constricting too much blood flow, but also having ice, putting some ice on the next to the.
All right. So let's get into the research. And that's, I don't know how far we are into the show, but that's why we have the doctor on the show.
Yeah.
It's in our wheelhouse. He's a positive Pacific Islander role model. But specifically the work he's doing, the scientific work that he's doing. We've had him on Koviki Talk, but now we are right in his wheelhouse. So go ahead and kind of walk us through maybe why you were looking in the first place, and then the result of the research.
Yeah, so this is the research we'll talk about. This was conducted during my postdoc at Genentech. So this was a 4-year training period where essentially after your PhD, you do a postdoc as a way to become an independent scientist in training. Right? So you have the resources given to you, and you sort of determine what kind of experiments you need to do to address the question. And so if you don't mind, can I just share, I think it would be nice to show you some of the highlight figures from the.
Yeah, let me do the multiple share. You should be able to share now.
Okay. That way you can get.
There you go.
Okay, so, all right, you can see the slides, right?
Yep.
Okay, perfect. So this is the paper that I just published. So you can see my name there. So I was the first author. So I was a co-first author with fellow postdoc Sterling Eckert.
And for those of you that listen to the podcast later and aren't able to see the screen, Dr. Al Young's name is first. And I don't know if that means anything, but it means something to me. His name is first in line.
Thank you. So usually at the first person is the one who led the project in terms of the experiments and thinking about the process. Everyone else on the list also contributed a lot to the project. So there are many people that can help you do many different things. So this was a very complex project. It involved many different techniques. And that just, that's goes to say to the value of collaboration in science, right? I can't do it all, right? I can't do the things that, other people can do. So you determine what question you need to answer and whether you have the potential to answer it, or you find someone else who's an expert. And I would rather give it to someone who knows how to do it than try to figure it myself and do it half-assed, right?
Right.
because I was already busy with so many other things. And so, and then if you notice at the end, so the end is usually my mentors, right? They're the lab that I work in. They're the one that provided all types of guidance to how, they still educated me on how to think about this. Because, as previously mentioned, when I did my PhD, I was doing it in biochemistry and structural biology, which is a completely different field from what I studied as a postdoc. The postdoc is where I got into more immunology, into more disease. But my PhD was more inside the cell, what is going on inside the cell. As a postdoc, I look beyond the cell, what is the output of what a cell does that affects disease, right? So that was also for me a strategic move because I wanted to, now that I have an idea of what's going on in the cell, I wanted to see how it translates to disease. And so, and I needed to get that training in order for me to like think about that kind of stuff. So that's sort of the layout of the authorship. And the title, as you see here, is Neutrophil Serine Protease 4, or we call it for short NSP4. And this is required for vascular leakage. This is required for inflammation. And required is a keyword here because that says that it's it, right? That's the gene that if you don't have the gene, you don't have inflammation. There are many genes that are involved in inflammation. So this is one of the genes that we have found that, and I'll show you some data in a little bit, that if we delete this gene from animals, so in our case, if we delete it from mice, these mice had zero inflammation. That's sort of the crux of the discovery. They had no inflammation, and we've looked at different disease models. So we've looked at models of rheumatoid arthritis. We looked at models of skin inflammation. So if you have like an allergic reaction, say if you get bitten by a bee and you should get swelling. If you have allergies to food, where you get hives. So hives is uncontrolled inflammation. And these animals do have zero inflammation.
So quick question.
Yeah.
And very general question. We hear a lot of tests and trials on mice. They're mammals. There's similarities, but are there more similarities, or why is it always the term lab mice? We hear that a lot. Why is it mice?
It's mice because it's so mice essentially have a lot of the genes that make up humans are conserved in mice. Okay, so they function the same way in mice. So mice are they're a convenient model that we can study. And mice also, we can induce the development of human diseases in mice, right? So like arthritis, we can induce the formation of arthritis in mice.
Gotcha.
So for example, this gene here, neutrophil serine protease 4, NSP4, we have that gene. And mice also have that gene. And so the idea here is what we found in mice is that if we remove the gene or if we alter the gene, it protects mice from getting disease, from getting arthritis, from getting inflammation. So the hypothesis is that perhaps the same gene works the same way in humans, right? So, and we're not just going to go and delete NSP4 from you or me, right?
Right.
FDA is not going to allow that. But we can develop drugs that can perhaps target the function of the gene, so not to delete it, but to block the function. And perhaps by doing that, we can protect ourselves from developing arthritis or whatever, some model of inflammation. And so the second important thing on the title here is mast cell, right? It's required for mast cell dependent vascular leakage. So it's required for inflammation that is driven by mast cells. And so mast cells are cells that are found everywhere in your body. And you're probably familiar with histamine. So, when you take antihistamines, so antihistamines is a way to block inflammation, right? So histamine, mast cells is the main cell in the body that produces histamine. Okay, so it produces histamine and produces serotonin. And these are essentially small molecules. So if you look at a picture of a mast cell, and I can show you a very cool picture here. I remember when I first saw this, when I first got this picture, I posted it on the Facebook. Um, People were so excited about it. Let's see. It's a bit slow. But basically, these cells are filled with pockets. So they have these granules, so these spherical features. And so the granules, maybe the image is not here. Yeah, so basically the granules are filled with histamine. Okay, so they're filled with histamine and different proteases and serotonin. So as soon as mast cells are activated, so here's an example of, it's kind of hard to see, but this is what I'm showing. Can you see my mouse?
Yep.
Okay, so what I'm showing here, this is the skin of mice. Okay, and you can see here this dark purple ones here.
Uh-huh.
These are mast cells. I actually had a better image that showed you exactly. Oh, here it is. Here it is. So this is an electron microscopic image of mast cells. So basically, this is really at the level of the cell, right? So this is one cell, and you see all the dark spots in there.
Yeah.
Those are granules, and those granules are filled with histamine and proteases. So when this cell, this is a mast cell, when a mast cell gets activated, right? If it sees like a food allergen that you're allergic to, right? It activates it and then releases. So basically, it releases all the histamine from these granules, and the histamine is a vasodilator. So it dilates your blood, and it like basically disrupts your blood vessels, and that's when you get the immediate. And this is why when you get an allergic reaction, it's very immediate, right? Like you have hives already, and that's because all these granules have high levels of histamine. And so when you activate these cells, they release it. You have inflammation. You have a hard time breathing because there's so much inflammation going on. So that's what a mast cell is. So basically, the gene that we discovered, it's important for how mast cells regulate inflammation. Okay.
Yep.
And so what let me just hop down to a specific piece of data. This is where this was the biggest discovery. So what we did here, so let me just take a step back a little. So when you think of rheumatoid arthritis animals, right? So this is, so we can get the features that we see in humans, right? We can get, sorry, can you still see me? My connection is a bit unstable, but you can see me, right?
Yeah, I can.
Okay, so you can see here the paw of the mouse, right? We can have the inflammation there. And so we can study the processes that are going on. So in the acute phase, this is when mast cells and neutrophils get activated, right? When they see like a food allergen, these cells get activated. They release histamine and serotonin, and that breaks apart the blood vessel indicated by these bars here. So it breaks apart the blood vessels, and you get now inflammation or influx of different molecules in there. And then the effector phase is that where you activate many other cells like T cells and B cells, so they come in and they make the process worse. Okay. And so what we found is that, so usually there's so NSP4 is the name of the protein, and then there's also a gene name, right? So there's a gene name associated with that protein. So the gene name for NSP4 is PRSS57. This is the gene that we all have. And this here, we're looking at the development of inflammation in these mice. So in a model of arthritis. So what we did here, so this at the very top, this is a normal mouse. We can induce arthritis, and you can see here that it over time, so over a period of 20 minutes, these mice develop vascular leakage. So we can monitor this. It's kind of like monitoring with like an X-ray. So we have an instrument to monitor a vascular leakage. And then if you look at the very bottom, these are mice where we remove the gene. So if you delete the gene, you can see here that it's completely gone. There's no inflammation whatsoever. And we also check for, we check for the swelling that we see in this example here, and there was no swelling at all. So these mice were completely fine. They had no response to the induction of arthritis. If you look in the middle here, this is another gene within the same family as NSP4. And so this here was a specificity test. Is the deletion here, is that specific to NSP4? Or let's say if we delete another member of the family, do we also see the same protection? And as you can see here, that's not the case, right? So it's very specific to NSP4. So this was the first big, aha moment for us that, okay, there's something really interesting about this gene. Any questions? Do you want me to clarify anything?
We have a question from Facebook
Okay.
related to inflammation, but the question is, what is your take on the Samoan Fulfo massage to reduce inflammation?
That's, it works, right? So massaging, it's a way of spreading sort of the inflammation. So it's not so much that it gets localized. So, but it's to spread it such that it doesn't, it's not to spread it to other parts, but it's just to control sort of the blood flow to that local site. So one example, one recent example is, like I said, my mom had knee replacement surgery, so she's going through physical therapy right now, and she had some swelling right at the local site. So what the therapist does is, in addition to like teaching her how to walk properly, is he massages that site to sort of like spread out, to not have too much inflammation in one side, so that it starts to develop, necrosis or some necrotic tissues. So yeah, no, for Samoa is if you need to get one, you should definitely get one for.
There's a bunch of people listening right now. They're going, "That's right."
Okay, yeah. I mean, we have a lot of traditional medicine that were established throughout the years, right? And it's through trial and error, and that's how science works, right? Like, there's a reason certain things exist today. And that's because over the years they've proven to work, right? Locally, just because we don't have access to medicine, and we have access to medicinal plants that our ancestors have used. And they've used it many years, and the methods that work have been passed down, right? So I wouldn't disregard those. Yeah.
We did an interview with an elder maybe a month and a half ago, and we had asked, what was healthcare like in Samoa growing up, and he said, it was like aspirin. We had aspirin, and then it was indigenous. You know what I mean? It was traditional Samoan practices, but that was it. And he just kind of made the comment that you dealt with whatever you had and with the tools that you had, and it was aspirin. And the practices that they had, but there was no true medical care in the sense that we think about it today.
Yeah.
There was no scientist looking for proteins.
Yeah.
Okay, so the bottom would be with the introduction of the regulation.
Yeah, so the bottom here is where this was the eureka moment that we thought, okay, there's something interesting about this gene. And so this is a full deletion, right? So when we make a deletion in animals, in this example here, we deleted the gene from basically every cell in the body. So there's no gene expression, and it's a full body deletion. So what we wanted to see next is what cell, there are many different cells in the body, and what cell in particular is contributing to inflammation. So what we did here is we systematically take different immune cells. So we take neutrophils and we put it back we take neutrophils that express the gene and we put it back into the mouse that doesn't have the gene. Okay. So these are mice that don't have disease. And we ask the question, if we put back one by one cells that have the gene, is it going to restore disease? Okay. So this is what I talked to you earlier about, like it's a puzzle, right? Like, and this is the part that I like about it, about science, about research, is you do very controlled experiments. And I don't know if you think that's an obvious experiment, right? It's pretty, to me, it's like an obvious experiment to like, okay, let's go one by one and see which cell is affected. So
it's
we
kind
did that.
of like a recipe, right? If you
Yeah,
change more than one ingredient at a time, you
yeah,
don't know why it tastes the way it does. If you
yeah,
do, like, let me just take one out or add one in,
yeah,
then you
exactly.
know that's why it tastes the way that it
Yeah,
tastes. Of course.
yeah.
So for Naki, we went, I don't know how far we are into the show. We're an hour into the show before we got to
Oh,
food.
okay, let me just wrap up then and show you the
Oh, you're
so
good. You're good. You're
yeah, am I good? Okay,
yeah.
so let me just show you this cool experiment. So here, okay, so this is a mouse that has the gene NSP4, okay?
Okay.
And when we induce disease, this is another imaging technique. You can see inflammation now, right? And all the
Ah,
pause, right? You can see this is inflammation, and we can monitor inflammation very easily. These are established protocols. Now, when we look at in this one and this one, these are animals that no longer have the gene, the NSP4 gene. And you can see here, you don't see the redness here, right? So these animals don't have inflammation. So what we did here is then we added in neutrophils. So neutrophils are also cells that are important for inflammation. So we didn't just add in any cell, right? We targeted the cells that had known roles in inflammation. So we added neutrophils, and the middle one are neutrophils that have the gene NSP4. When we added that in, we didn't see that they now became inflammatory, right? They still didn't have any signal. And as a control, right, we added neutrophils that also don't have the gene NSP4. And again, they don't have the same response as these ones. So then we looked at another cell. We looked at mast cells because we knew mast cells also played a role in inflammation. Again, here on this side here, this is the normal mouse that has a gene, and they have inflammation. When we look at the mouse without the gene, where we've added in mast cells that have the gene, you see now what happens is they get inflammation back. But if we add in mast cells that don't have the gene, they don't have inflammation. So they have mast cells, but they don't have inflammation. And that was the definitive, another eureka moment where we found not only that the gene is important for inflammation, but we found the cell type that gives rise to this inflammation. Okay, so we have the gene inside the cell that's regulating something inside the cell, and that's affecting that ability of that cell to give rise to inflammation. And so we go step by step, right? So now we've identified the cell, then we look inside the cell. What is the gene doing inside the cell that regulates inflammation? And what we found in a series of other experiments is histamine. So when we knock out the gene, we reduce the level of histamine in the animals. And that's why when the animals, and that's why in the animals, I think I'm missing one piece of data. But anyways, for example, here, so in this, if you just focus on this one experiment here, and here we take mast cells that have the gene and don't have the gene. The ones that have the gene are blue, and the one in no gene is red. And we treat them with an agent to have them get activated and release histamine. And then we measure how much histamine is being released. And as you can see, just by the look of this, we have more histamine in the blue and then less histamine in the red. Okay. And histamine And what we did, and I don't have that piece of data here, is that when we take the mice that don't have inflammation and we simply inject histamine, that bypasses the mast cells. These mice develop inflammation because now histamine is released, right? So essentially, the discovery is that we found a gene that regulates histamine levels inside mast cells. When we knock out the gene, histamine levels go down, and you know histamine is important for inflammation, right? And when histamine levels go down, these animals are protected from developing inflammation. So when you think about drug development, the idea is, can we make drugs that target this gene such that it blocks their ability to regulate histamine, and as a result, you reduce inflammation. I hope that makes sense.
It does. So there's is there the same amount of histamine in play, I guess, just is not released, or does the cell create histamine and release it?
That's good. So what happens is when we delete the gene, the cell makes less histamine.
Okay.
Okay. So the normal cell and the cell without the gene, they have the same potential or the same ability to release, right? So the releasing process is not affected. So that was actually another point that we looked at. Is their ability to release things, is that affected? But no, that wasn't affected because we looked at other things other than histamine that are also stored in the granules, and they had same levels and they were released the same way. So it's really the histamine levels that the cell is making is much less. So they were making less histamine.
You've really simplified I'm sure how long was the process? How long was from beginning of like, we're gonna try to delve into this to the creation of this?
So this was a long process. I mean, it took 4 years of my postdoc, right? But there were, making the mouse that doesn't have the gene had started before I started the postdoc, right? So the earlier work was, when I started, we already had the mouse available, so I was able to just go right in and do experiments. But it was a long process, and that's typically how science works, right? So I would say like at least 6 years.
Wow.
But then throughout my postdoc, then there's the review process, right? So I don't just publish, I don't just finish my work and then get this out. So trying like getting the publication out, this took a year. So I left after my postdoc while I was still working on getting the paper reviewed. So this paper was sent to many different reviewers, experts who have not worked on it, and they tell us whether this is worth publishing or not. And they review the data, and they've asked us for like, okay, you need to do more experiments. So I was in my second job while I was still going back to Genentech to finish up, do more experiments, because that's how the process works. You don't just publish anything. You have to go through a very serious review with people who may agree or disagree. And some people fail the whole review process because there's someone on the committee who's like, no, I don't believe this. So it's a brutal process, and I'm glad it's over. I'm so glad it's out now.
It's like well, we don't have babies, we're men, but it's like, I can imagine there's a labor process. Now there's a baby and I can move on.
Oh yeah, that's exactly, this is my baby. It's out now.
Well, I can see that just understanding the importance of inflammation and how it aggravates a whole bunch of other things going on and takes energy away from the body trying to heal, other parts of it. This will be a big deal in the medical field. And I'm going to think of you every time I take an antihistamine. I'm going to think about our conversation.
It's targeting mast cells. Think about mast cells.
Yeah, I'm gonna use that term, mast cells, with my wife. She's a She's got a genetics degree, so I'll throw that out there, and she'll go, "Who you been talking to?"
There you go. There you go.
Well, doctor, it's been a pleasure talking to you. Is there any information or anything you want to get out before we wrap it up? And you weren't just a guest today; you were kind of the co-host guest as well. So you played a couple roles today.
I hope I've expressed, my love for research. Like I love what I do, and I hope that young Pacific Islanders who are seeing this, Samoan, Tongan, Hawaiian, Tahitian. This is, think of it as like your video games, right? I know there are a lot of like video game lovers. It's strategizing, coming up with, different plots, and this is my video game, right? So, and you can do the same. And there are these moments like the discovery that you completely block information that just makes you want to keep doing this, right? And there are scientists who go through the process and they don't have as many eureka moments, so they decide to take other careers. But for me, I've been so blessed and so lucky to have from the beginning, family who supported me, my parents who supported me, I had teachers who supported me, I had mentors throughout grad school and postdoc who supported me. And I was getting these eureka moments that kept me going, right? So I hope that you guys can give it a try. Just ignore all the memorization that you learn in school, but like research is really, it's different, right? It's where you actually you decide how you wanna play the game, and that's and I hope we get more representation in these fields. So that's the message that I wanna get across. But also to thank you and Naki for the platform, for inviting me to share this work, and it has relevance to all of us, I believe.
Well, thank you, doctor. And that's a great message to send out there. Like, look, we're all going to work hard in our lives. Look here, we have a doctor who's doing what he loves and working hard. The hours will seem shorter when you're doing what you love. And when you do what you love and you're tired at the end of the day and long hours and those kind of things, I can imagine it's a good tired.
Exactly. Yeah.
That's much different than doing things you don't love to do. So,
especially when it gets published.
Great, exactly. Especially if it gets put out there for everybody to see. Okay, well, thank you so much. We're going to give you the rest of your Sunday back. On behalf of Naki, I'm sure that actually she's messaging me and making sure that we're following the right guidelines. So she's still here. Thoughts and prayers to the Moley family as they are giving COVID some dirty whoopings right now. They're going to be done soon. Thank you, Dr. Ai Young, and enjoy the rest of your Sunday.
Yeah, you too. All right.
Love you.
Take care. Bye.